DNA replication is nature's ultimate copying machine. It unzips and perfectly copies billions of genetic letters in minutes. It's how life grows, heals, and passes on traits. Basically, it's the secret code that keeps every living thing running. DNA replication is when the two strands of DNA, made of the four chemical bases A, T, C, and G, are twisted together in a double helix shape. They start to separate with the help of an enzyme called helicase. When the two strands start to separate, it forms what's called a replication fork. Now, before moving on, it's important to note that the two strands are complementary. That means if one strand has an A, the other will have a T, and if one of a C, the other will have a G, and vice versa. Starting with the leading strand, which runs from the 5' to the 3', an enzyme called primase adds a RNA primer onto the strand to get the replication started. Again, it's important to note here that instead of using the complementary base T for A, it instead uses a U, since RNA does not contain a T. Once the primer is placed, a DNA polymerase binds to the primer and goes down the line, attaching complementary DNA bases onto the DNA until it hits the end. Remember, the DNA polymerase can only move from the 5' to the 3' end. Because of that, the leading strand is made continuously and does not need to break. In the lagging strand, because it doesn't move 5' to 3', the primase will place primers all throughout the strand, and the polymerase will attach DNA from primer to primer, creating Okazaki fragments. Once we finish making DNA, the enzyme exonuclease comes and removes the RNA primers from the strands, and another DNA polymerase comes and replaces it with the DNA. Once that is complete, ligase comes and checks both the strands and seals them shut. Because DNA polymerase has a proofreading system, this limits the amount of errors that occur during replication, but do not stop it from happening completely. Most commonly, an error occurs in placing the bases, and though some are harmless, if not detected, this can lead to segments of DNA being deleted, changed, or added on, which can further lead to mutations in the DNA. When DNA polymerase finds the incorrect base, the exonuclease comes in and removes the incorrect base, and the DNA polymerase comes back and adds in the new one. And then, once that is done, it continues with the DNA replication. Because DNA is conservative, each time it replicates, there will always be one old strand and one new strand. And in this image, you can see how it's done throughout 4 different generations of DNA.